Hello everyone again. Good morning, good afternoon, good evening, and good early morning to US. We have a lot of participants again from all over the world. Right now we have 186, 87, 189 participants from different parts of the world. We are asked to give some numbers about what happened yesterday. We had about 550 unique views, and we had a range of number of participants from 250 to 360 throughout yesterday. So we had a bit of fluctuation. People came in and they left. And we had a very successful day yesterday. All the recordings went well, and I think the technology also was in our favor. I have uploaded the videos to YouTube. I'm waiting for the speakers to give me their confirmation that they're happy for me to release the videos. Hopefully throughout the day, I will put the link to the playlist in the slides so that you can already view the ones that you want to view from yesterday. And the videos that we record today, we are going to put it online. Hopefully tonight, they need a bit of processing, and hopefully by the end of the week, like by the end of Friday, you will be able to access the videos for today. So without any further ado, I would like to introduce our very first speaker today. Our first invited speaker is Professor Stefan Koelsch from the University of Bergen in Norway. He is the leader of the Brain and Music group located at the Institute of Biological and Medical Psychology, and has a keen interest in investigating how the brain is affected by music. Especially, Stefan has researched into the neural correlates of music emotions, the neurocognition of music, and music therapy, with many published papers in prestigious journals such as Nature Neuroscience. It is with these insights that Stefan and his group try to understand how music can help ease symptoms and decrease illness severity in cases of Alzheimer's, cognitive impairment, and cognitive decline. Thanks a lot, Stefan.
Yeah, thanks, Amir, for organizing this and for inviting me. I'm very happy about the opportunity to share thoughts and research in this webinar. And I'm going to try to share my screen now. And I hope you all are able to see my presentation now.
Yes, it is visible now.
Great. Yeah, I'm going to talk about brain correlates of music-evoked emotions. And I wanted to start talking about how I investigate them with music and an illustration of emotional responses in the brainstem. So auditory information can evoke visceromotor or autonomic responses via the so-called inferior colliculus, schematically illustrated here. And the inferior colliculus is part of the brainstem, which you see here. In addition, the vestibular nerve also contains a substantial number of acoustically responsive fibers, and the vestibular nuclear complex exerts influence on ocular motor neurons and spinal motor neurons in response to sounds, in particular these sounds with low frequencies and with sudden onsets. So for example, when there's a loud bang, then you might startle, your eyes shift direction, there might be an increase in heart rate, breathing rate changes, sweat production changes, and so forth. These are both reactions of the somatic motor system and the visceromotor system in response to sounds. Also, both the vestibular nuclei and the auditory cochlear nuclei project to the reticular formation, which is not shown here. And the vestibular nucleus also projects to the parabrachial nucleus, also not shown here, which is a convergence site for vestibular, visceral, and autonomic processing. And such projections initiate and support movements, and they contribute to the arousing effects of music, or also to the calming effects of music. And I wanted to share this with you here because it illustrates that even the earliest auditory processing stages give rise to effective responses to music. And that is also why I like to speak of an auditory limbic pathway, which is shown here in light blue, which then also includes the medial geniculate body of the thalamus, the auditory cortex here, and the amygdala. Now, beyond the phylogenetically old brainstem systems, which are in part the basis for our visceral reactions to music, several forebrain systems also contribute to emotional experiences. And this is illustrated here. These are results of a meta-analysis of a number of functional neuroimaging studies illustrating several neural correlates of emotions evoked with music. And the studies included in this analysis used various experimental approaches, for example, investigating emotional responses to consonant or dissonant music, music sounding happy or sounding sad, music that evoked joy or fear, musical expectancy violations, musical tension, and also music evoked chills or musical frissons. Now, this meta-analysis indicates clusters of activity changes reported across studies in the amygdala, with a local maximum in the left superficial amygdala, that's indicated, outlined here by the green line, and in the right laterobasal amygdala, which is outlined here in red. Also, the cluster extended into the hippocampal formation, including the cornu ammonis of the hippocampus and partly also the subiculum of the hippocampal formation. Then there were other structures such as clusters in the striatum. Here we see the left superior or dorsal striatum and the right ventral or inferior striatum. This cluster here includes a structure that we refer to as the nucleus accumbens. Then here in panel C, we see clusters in the orbitofrontal cortex, in the cingulate cortex, in the thalamus. In panel D here, we see auditory regions of the superior temporal gyrus, including Heschel's gyrus and a more anterior portion of the superior temporal gyrus. There was also an activation in the parietal operculum, which is not shown here. This region includes secondary somatosensory cortex. And there was a small cluster in the posterior insula, which is also not shown here. Now, I will not discuss all of these clusters now, but only make a few remarks on some of these clusters. So first, with regard to the nucleus accumbens, this structure, the nucleus accumbens, is sensitive to what's referred to in the psychological literature as primary rewards, such as food or drink or sex, and secondary rewards, for example, money or power. And the nucleus accumbens is involved in representing hedonic value, and it motivates, it initiates, and it invigorates behaviors that are aimed at obtaining rewards and at consuming rewards. And therefore, I also refer to the nucleus accumbens, figuratively speaking, as the fun motor. It's important to consider, I think, that the nucleus accumbens is also, if not first and foremost, a motor structure. In addition to the cluster in the right ventral striatum here, the meta-analysis also indicated this cluster in the left dorsal striatum, the caudate nucleus, and the pattern of this co-activation of these two structures here corresponds exactly to activations in response to food, money, or erotic rewards. This here is from another meta-analysis. This meta-analysis is by a colleague, Guillaume Siscous, and it's on the processing of primary and secondary rewards. So let me go back again so that you can compare these two clusters here with the clusters in the previous slide, evoked by music, evoked emotions here. Now we know that such activations in response to monetary, food, and erotic rewards are to a substantial amount due to dopaminergic activity. So dopamine is one of the neurotransmitters in the brain. And here is one of my favorite neuroscience studies on music and emotion by Valerie Salimpour from Robert Sartori's group. They investigated neural correlates of music-evoked emotions, more specifically of musical pleasure or peak experiences of musical pleasure, so-called chills or frissons, that is, intensely pleasurable responses to music that often involve, for example, goosebumps, shiver down the neck, shivers down the spine, etc. This was a PET experiment in which they used a tracer called Reclopride to measure dopamine availability. And the method was that each participant brought a piece of music into the lab to which he or she would experience such a chill. And as a control stimulus, that person listened to the music that another subject brought into the lab. So the authors report that during listening to music that evokes chills, they observed increased dopamine availability in the caudate, that's shown at the top panel, in the putamen, and critically in the nucleus accumbens, that's indicated by the white arrows in the bottom panel. And this shows us that music-evoked pleasure involves dopamine release into the striatum, that is, activation of the dopaminergic mesolimbic reward pathway. Now, the finding that musical pleasure involves dopamine has then been confirmed by other studies. For example, this study by Laura Ferrari, whom I saw yesterday also in the list of participants. So hello, Laura. In this study, availability of dopamine was modulated by administration of levodopa or risperidone, and it was found that such modulation results in higher or lower pleasure ratings of music. Now, let me go back to this meta-analysis on reward. I've mentioned these striatal activations here. In addition to these activations, the authors reported that primary and secondary rewards also engage a number of other structures, specifically the ventromedial orbitofrontal cortex, cingulate cortex, amygdala, anterior insula, and mediodorsal thalamus. That is, reward is not only processed in the fun motor, the nucleus accumbens, this structure here, but it involves an extensive network in the brain. And strikingly, this entire network shows up in this meta-analysis on music-evoked emotions, including orbitofrontal cortex here, and cingulate cortex, amygdala, insula, and also mediodorsal thalamus. And this indicates that much of what we see in this analysis is associated with the experience of music-evoked pleasure. Now, interestingly, this pleasure network overlaps substantially with the pain network. Here's an illustration of the pain network on the left and a different view of the meta-analysis on music-evoked emotions on the right. And here we can see the overlap of the pain network with the pleasure network, including structures that I just mentioned in the prefrontal cortex, amygdala, in the striatum, insula, mediodorsal thalamus, cingulate cortex. In the beginning of my talk, I mentioned the parabrachial nucleus, and here also secondary somatosensory cortex, which we see for music-evoked emotions in this view here. And it's probably due to this overlap that music can modulate pain. This here is a meta-analysis that reported that listening to music reduces pain intensity levels and opioid requirements. However, this analysis also concluded that the magnitude of these benefits was relatively small compared to pharmacological drugs. But maybe there are certain more focused or more attentive listening techniques to music or other forms of engagement with music that might actually enhance the pain-reducing effects of music. That's, I think, an interesting question for future research that we also currently address in my research group. So coming back to the meta-analysis on music-evoked emotions, I've already said that much of what we see in this meta-analysis is associated with the experience of music-evoked pleasure. So such pleasure might arise, for example, from the experience of beauty, from emotion regulation, from triggering pleasurable memories, and from social bonding. And this is where perhaps the most interesting finding of this analysis comes into play, namely these clusters here, indicated by the yellow arrows. An overwhelming proportion of studies on music-evoked emotions have reported activity changes within the anterior hippocampal formation. And this stays in striking contrast to monetary or food-related or erotic rewards, which do not activate the hippocampus, as for example evident from the meta-analysis on reward processing. Now, activation of the anterior hippocampus is a very interesting capacity of music, and it indicates that music-evoked emotions are not related to reward alone. There is more. And in the following, I will argue that music easily stimulates social bonding and evokes attachment-related emotions, and that such attachment-related emotions are generated in the hippocampus. This here is from a review by Brian Strange, who shows that the hippocampal formation has, on the one hand, dense bidirectional connections with the amygdala, that's shown here on the right, but also with the nucleus accumbens, shown here in the middle, and the hypothalamus, that's shown here on the left. Now, interestingly, is particularly the anterior hippocampus projects to the so-called hypothalamic medial zone nuclei. That's indicated here. And these nuclei form circuits involved not only in integrating innate defensive responses to, for example, environmental threats, but also in mediating reproductive and social behaviors. So these nuclei mediate release of oxytocin, and in addition, hippocampal hypothalamic projections also regulate social bonding via a range of other neurochemicals, among them endogenous opioids. I'm going to come back to the endogenous opioids a little bit later. First, I wanted to mention that in the course of evolution, the hippocampus, which is shown here in blue, was later differentiated than the diencephalon, shown here in green, with the thalamus and the hypothalamus, and later differentiated than the brainstem, shown here in yellowish. Now, mammals and birds need to nurse and to care for the offspring. That is, they need to feel attached to the offspring, to recognize the offspring, and they need to be able to remember the way back to the nest with the offspring. And all of these functions appear to be co-localized in the hippocampus. So social attachment, navigation, memory, etc. And these pieces of evidence led us to the notion of a hippocampus-centered affect system, which generates attachment-related emotions and affects associated with the care system in the brain. Now, in humans, attachment-related emotions are associated with feelings such as tenderness, for example, sympathy, compassion, peacefulness, kindness, and the feeling of being moved that David talked about yesterday. We associate these emotions with joy and with happiness. And on the other hand, when social attachments are severed, it can lead to, for example, sadness, grief, or unhappiness. Now, as I've said before, the hippocampus has direct projections into the nucleus accumbens, and that is why joy and happiness can also be fun. But not all fun is automatically including, joy or happiness. I can say that, for example, ice cream, chocolate, money, big cars, etc. are lots of fun. They don't make happy though. Whereas things that make happy, for example, making music and dancing together with people that you like, being compassionate and kind, etc. These are things that are also, fun. Well, now, assuming that the hippocampus plays a role in attachment-related emotions, one important link to music comes into play. And that is that music is special in how it supports and promotes social functions, such as contact between individuals, communication, cooperation, thus promoting social attachments and also social cohesion. Now, engaging in these social functions is a human need. The social psychologist Burmeister talked about what he called the need to belong. And the fulfillment of this need is perceived as pleasurable and as something that makes us happy. On the other hand, exclusion from such social functions, on the other hand, makes us unhappy. It has negative health effects and leads to increased mortality. That's also why, in these strange times where the word of social distancing goes around, it's often forgotten that social distancing is actually something that's harmful for us. Physical distancing, yes, but social distancing is something that we should actually not engage in. And I mean, let's not forget that in order to socially connect, we do not necessarily have to be always in the same room physically close to somebody else. Now, coming back to music and emotions again, what I wanted to highlight here is that synchronization between individuals appears to promote social bonding. And that includes emotional synchronization, or copathy, as I like to refer to it, and synchronization of movements. That is, movement coordination between individuals. Here's a model by Robin Dunbar and colleagues, making a strong connection between synchronization to music and social bonding. Now, synchronization of movements between humans leads to increased trust, increased cooperation, and thus also increased social bonding. This review here by Dunbar, Tarr, and colleagues provides a very nice overview about the studies showing this. And the authors of this article here also make a strong point that the endogenous opioid system is activated by synchronization. And this endogenous opioid system is also involved in social bonding, as I've mentioned before. It is also involved in the reduction of pain and the reduction of anxiety. Now, the authors here argue that endogenous opioids, and particularly endorphins, play a central role in the establishment and maintenance of non-sexual, non-kinship social bonds, and that particularly endorphins are important candidates for the neurohormonal underpinnings of social bonding during group musical activities. And the activation of the endogenous opioid system with music is then also associated with pain reduction and reduction of anxiety. I wanted to highlight this here because the ability to synchronize movements, including vocalizations, flexibly in a group to an external pulse, the tactus or the beat of the music, is uniquely human. No other species can do this. And I believe that this ability is the simplest cognitive function that just separates us from animals. And this would make music the decisive evolutionary step of the Homo sapiens, maybe even of the genus Homo. So I think the step from not being able to synchronize movements flexibly in a group to an external pulse to this ability is the step from the animal kingdom to humans. So gorillas, for example, yes, they drum and they pound on their chest, and they do this even in groups, but they do not do this in synchrony. Or whales, yes, they sing, and they sometimes even sing with more than one individual, but they do not sing in choirs as we humans do. So music usually has a pulse and a scale, and that is what makes music special. And by virtue of these two fundamental musical principles, pulse and scale, music immediately affords that several individuals produce sounds together. That's what pulse and scale are for, that we can easily produce sounds together. And this is in contrast to language, which also has many musical features, such as speech, melody, timbre, rhythm, etc. But language is best understood when produced by one individual at a time. Only one person can talk at the same time, otherwise it doesn't sound good and we cannot understand anything. And with music, several persons can produce sounds at the same time and it still sounds good. Okay, one can still understand the music. So language was evolutionarily adaptive because individuals could communicate what they think, what they want, what moves them, etc. And music was evolutionarily adaptive because it brought the capacity to establish and support group cohesion in big groups. And therefore, I believe that without music, humankind would actually not have made it through evolution. So next, I will talk a little bit about brain correlates of emotions evoked by musical expectancies. And the main reason to focus on this is to connect it to Marcus' talk from yesterday and David Huron's work on anticipation in music. So this here is a bigram table, a table with chord transition probabilities extracted from a corpus or computed for a corpus of Beethoven string quartets. This is from Fabian Moss and from the group of Martin Rohrmeier in Lausanne. Now, I'm going to mention a few things that Marcus already mentioned yesterday, and I'll try to illustrate them using this bigram table. So chord transition probability means that it shows us the likelihood for the transition of one chord function, shown here in the left column, to another chord function, shown here in the bottom row. So for example, after a dominant seventh chord, there is a very high likelihood that the next following chord is going to be a tonic. This is indicated by this upper red arrow here. And let's say during the submediant here, for example, well, we can see none of the chords to follow is really highly likely. Maybe the supertonic here, which has a little bit, a bit higher percentage than the other chord transitions. Now, what this means is that during a dominant seventh chord, we have a relatively high certainty that the next chord is going to be a tonic. And when I say we, then I mean all the individuals who have had, lots of exposure to major, minor, tonal music, and therefore implicitly at least acquired all these chord transition probabilities, more or less, that you see in this table here. Now you might ask, isn't that totally different for other composers, other musical styles, etc.? Not really. We did a similar analysis also, or Martin did a similar analysis also with the corpus of Bach chorales. Turns out the basic principles are surprisingly similar. So, during the presentation, during the listening to submediant, so the 6 here, we cannot really be certain about which chord comes next. Maybe, maybe we expect this one here more than the others, but the level of uncertainty will be higher than during the dominant seventh chord, okay? And by applying the formula of Shannon entropy, we can, for this array here, compute this value here. So 0.37 for the dominant seventh chord and 0.59 for the submediant. And this entropy is inversely related to our certainty about what's going to happen next. Okay, so low entropy, we are pretty certain what happens, or we are the uncertainty is low, and during the submediant, uncertainty is relatively high. We are not certain what happens next. Also, when after the dominant seventh chord, the tonic does not appear, so when the next chord that we hear is actually, one of those guys here, then we will be surprised because we did not expect that chord. And Marcus already referred yesterday to a study in which we investigated expectancy, unexpectancy, certainty, uncertainty. It was a study from my PhD student at the Max Planck Institute in Leipzig, Vincent Chiong. And he said that in this study, instead of just using a chord transition probability table as shown in the last slide, we used Marcus information dynamics of music, the idiom model. Because that model does not only rely on the context of one chord, as in the bigram table on the last slide shown, but it actually takes into account several of the previous chords. It takes into account repetitions of certain sequences and many things more. It's a much more sophisticated model than just this bigram table here. So we used that model to determine for each and every chord how certain or uncertain participants would be about what the next chord would going to be. And for each chord that actually sounded, it provided a value of how surprised or not surprised the participants would be. This study was actually carried out with a corpus of pop music songs, not Beethoven string quartets. Also, we modified the stimuli or the songs a bit so that each chord would actually sound more than 2 seconds. And that was important because we also did functional MRI with this in order to investigate neural correlates of musical expectancy, surprise, uncertainty. So again, just as a reminder, during each chord, we have one value about how certain or uncertain participants would be about what the next chord is going to be. And also for each and every chord, we have a value about how surprising or expected this actual chord would be. Then in one experiment, we had participants rate the pleasantness of chords. They did this on this slider interface here. In other experiments, we also obtained ratings of surprise and certainty to validate the model. But now we are interested in the emotional effects of surprise and certainty with regard to musical pleasure. That's why we asked participants in this study to rate the pleasure for each chord so that we could analyze how certainty, uncertainty, and surprisal gives rise to musical pleasure. And what we found is, as Marcus already explained yesterday, that the interaction between uncertainty and surprise predicted pleasure ratings. So here, in panel B, pleasure is shown in red and displeasure is shown in blue. And in a situation of high uncertainty, on the right of the image, participants found it pleasurable when chords were actually not surprising. When chords were unsurprising and they were not certain what was about to come, they found surprising chords rather unpleasant. In other words, when I do not know the system, when I don't know where this is going, then I want chords to help me orient or to learn the system. And thus, other than regular events are confusing and unpleasant, okay? However, in a situation of low certainty, in the left side of the image, that is when participants think, I know what's coming up next. I know where this is going. Then participants found it pleasurable when chords were actually surprising. That's shown here in the top left quadrant. In other words, when I know the system or when I know where this is going, then I like it when I'm surprised. And this is also consistent with real music experience. Composers put unexpected events in positions where you already have a certain kind of idea what is coming up. And then they don't end the phrase, for example, or they don't let the phrase exactly go where you thought it might go, but make a small change, make a certain surprisal so that it doesn't get too boring. So in figure C here, which is the one that Marcus showed yesterday, we have a three-dimensional representation of our findings. And here we can see it's not just a sweet spot of the amount of surprise or of uncertainty as suggested in the inverted U-curve models, but it's a complex shape. You have to change multiple things, not just one. And then in this three-dimensional space, you get this ridge here, which resembles the inverted U shape. Yeah, just briefly touching upon some of the MRI findings. In the MRI analysis published in this paper, we did a region of interest analysis using the clusters that I presented before from the meta-analysis on brain correlates of music-evoked emotions. And for the amygdala hippocampus cluster, we see that it's actually not sensitive for the uncertainty, nor for the surprisal, but for the interaction, that is for the pleasure that arises from the interaction of uncertainty and surprise. And the auditory cortex shows responses to both the unexpectancy, the surprise, and the interaction. Now, what about the nucleus accumbens, which was one of the structures I was very interested in, because I thought, well, isn't it curious that the nucleus accumbens doesn't show up in our studies with expectancy violations in music, because unexpected chords help you to improve the model and should be therefore expected as rewarding. But usually, or never in these analyses, we saw the nucleus accumbens. So in this analysis, on this study here, it turns out that the nucleus accumbens is actually sensitive to the uncertainty. So the fun motor in the brain is, if you will, a kind of page turner in the brain. It motivates us to continue to listen to the music, especially in situations where we are uncertain as to how the current situation is going to be resolved, as to how the current harmonic configuration is going to be resolved. I just wanted to share one different approach to investigate this issue with functional MRI. And this here is from a study that I did together with Moritz Lehn a few years back. In this study, we wanted to investigate neural correlates of musical tension. We presented music pieces to participants and obtained continuous tension ratings. The gray lines are tension ratings of each single participant, and the black thick line is the average tension curve. And then we did an fMRI experiment in which we used the black tension curve as a continuous regressor in the fMRI analysis. Now, first I'm going to present you the sound example, or one sound example. This is one of the stimuli that had both, it was the natural performance by a pianist. However, we also had other versions where we removed variations in tempo, where we removed variations in loudness, or where we removed both variations in tempo and loudness. This is the one that is the natural musical performance. I will try to play you the beginning of this piece. It's the Venetian boat song from Felix Mendelssohn Bartholdy. we are around here in the curve, I guess. That's this here. Return to the tonic. This could be the end of the piece. So low tension ratings here. Increasing uncertainty. Surprising harmony. And so forth. So, as I've said, we then used this tension curve as a continuous regressor in an fMRI experiment. I should say that in the fMRI experiment, participants were not hinted at that we were investigating tension. They were just asked to lie in the scanner, relax, listen to the music, and after each piece, we obtained pleasure ratings of the pieces. They were not informed that this was actually an experiment on musical tension. And if I remember correctly, then half of them were non-musicians. So the findings that I am going to show you now is not just a finding for musicians. So in that study, what we found, I should also say that, of course, we used the acoustical regressors, in particular loudness, as additional covariates of no interest. We did the analysis also only for the deadpan versions. So what I show you here now is not just an effect of the music getting, faster and louder or slower and louder with increasing or decreasing tension. So using the tension curves as a continuous regressor revealed this spot here in the orbitofrontal cortex as correlating in its activity with the tension curves. And although I'm not going to show you all of the details of other studies now, but this is a region that is has often been reported in other studies on musical expectancy violations, where it shows activity changes, and also in the study that I did with Vincent Cheung and Marcus Peers, the one that I showed before. In the whole brain analysis, we also see exactly this spot, actually on both hemispheres there, showing us that what we find here for the tension ratings is indeed also related to uncertainty and surprisal as evoked with music. Yes, and this is my final slide. I put this up only to say two things. First, the studies cited here, this one and the next one, are studies in which we used EEG to investigate the processing of unexpected chords. And at the same time, I'm going to go back here, we also measured the skin conductance, so the sweat production on the palms of the hands, which may change as an autonomic effect of emotional processes. And I'm not going to go into details here. The only point I wanted to make here is that expected and unexpected chords also have peripheral physiological effects. Here, effects on the sweat production on the palms of our hands. That is, they have autonomic effects that change the activity in the organs of our body. And I'm just saying this because maybe for the next conference, Amir, when you want to add another circle, then you could add, the body as another circle, which is affected by the things that happen in our brain, which can be modulated by listening to music or by engaging with music. And with that, I would like to thank you very much for listening.
So great. Thanks a lot, Stefan. It was really interesting. So regarding the body and brain thing, yesterday we also had a comment on dualism, that we don't separate body and brain. So you're right, we might just make it body brain, like B in a way, or a fourth or a fifth circle to include body. So that was great. So we have about 9 minutes for the questions. I had to merge a few questions together. So let's begin with the first one. Has any of your research shown that music can inhibit communication across somatosensory areas of the brain, and by doing this reduce perception of pain or exertion during activities such as heavy labor or exercise? Or if you have examined the effect of listening to music on the reduction of inflammation in the body?
Short answer is no. I'm just searching through my brain which things of interest might be relevant here. So we've done one study in which we investigated the effects of happy sounding and sad sounding music on the so-called default network in the brain. And there we found that sad sounding music evokes more or less positive thoughts or more negative thoughts in some subjects, and at the same time increased activity in the default network, which again then is associated with certain functional connectivities among the structures in this network. But that would be an increased functional connectivity in response to sad sounding music. So that's not really what the question, that's not a yes to the question. And also I should add that there are, at least to my knowledge, no functional neuroimaging studies on pain modulation with music. Joke Bratt and I put in a grant last year to the NIH to investigate this, but unfortunately it was not funded.
Okay. Well, good to hear that some of the grant application of like big name like yours also get rejected for more early career or junior researchers. That's
More
very
often
interesting.
than I would like to admit.
Okay, actually in that regard, there is also one question: that does the music evoke emotions meta-analysis include like fearful or negative valence music as well, or no?
Yes, there were studies using music evoking fear, taken from horror movies, television series, or thriller series, ego shooter games, things like this. However, although in our own study we obtained valence ratings and found that the valence, so pleasure for the fear evoking music, was lower than for the happy sounding music, in real life or real aesthetic experience, etc., we don't really perceive it as negative. We find the thrill often positive. That's why some people, like to listen to watch horror movies, and they want the music with the horror movies to heighten the aesthetic experience of thrill.
Okay, yeah, that's very good. And there are also like multiple studies looking at these things.
Yes, it's also, there are several studies, two of them from my lab, fMRI studies with music something like happy and fearful. The whole field with, negative emotions and music is very complicated, because sad music is not necessarily negative or negatively valenced, same for fear evoking music, etc., etc. Yeah, but that's a different question.
Exactly, yeah. And I think David also talked about different aspects, that something that is sad might not be really sad. Yep, yeah, very good. Do you know of any ethnographic studies that have sought to confirm or deny that these ideas about synchronization and bonding reflect people's real experiences of participation in music?
Well, as far as we know, every culture has music, including dance and making music together, singing together, etc. However, I think it's important with which attitude participants engage in the social activity of musicking, if you will. Because in a situation where we do this with the orientation of peacefulness and kindness, the effects will be different than in situations where we do this with an attitude of aggression, for example, or ostracizing also. And I think this will both have very different effects. For example, I think that the true therapeutic effects of music will only unfold in a situation where music is experienced or made or practiced in a way that is not aggressive, but in a way that aims at social bonding in kind and peaceful ways.
Yep. One final question. Majority of clinical studies in pain management tends to describe and refer to music as a single intervention rather than a large group of heterogeneous interventions, which challenges the validity of music classification. So the person who asked this, as a music clinician and researcher, I was wondering if you could say something about elements of music besides preferred music, which are involved in music-induced anaplesia.
Yes, very good question. Very complicated question. I will try to keep it short. I've already said that one important aspect of music is the beat and the scale, which differentiates it just from sound. You know, fire crackling or breaking waves, etc., etc. We had this discussion yesterday after the very last talk. Now, if I have a beat, for example, I can engage in the beat. And engaging in a beat, synchronizing to a beat, even if it's just, you know, with the finger, let's say, does two things. First, it probably, if only ever so slightly, engages the endogenous opioid system with, you know, the cascade of neurochemical effects that I touched upon in my talk. And second, let's not forget that this also takes attention away from the procedure that evokes pain, from the pain evoking stimulus itself, from the worries associated with the pain, etc. And just directing attention away and on the music will probably have significant pain reduction and anxiety reduction, reducing effects.
Great. Thanks again, Stefan. I'm afraid we received 29 questions, but I could ask only a few.
Maybe one more.
One more, okay, sure.
It's 29. So we have time for one or two more.
Okay. You showed quite a lot of brain areas in response to music. Pretty much all those brain areas were also involved in primary and secondary reward system. What about in the reward system, like other, like food and stuff? What about the studies looking at causal link between these brain areas, like dynamic causal modeling? Would you think there is one or two central brain areas that initiate the activity of all the rest of the brain areas? Or is it a collective activity that leads us to perception of different emotions?
Yes, again, very good question. again, very short answer so that we can maybe even do one more. The study by Ferrari that I mentioned kind of goes into that direction with dopamine modulation. So that there you have kind of a causal link. And then please also look up a study from Robert Tsotori's group where they used transcranial magnetic stimulation, TMS, to modulate frontal areas that talk to areas with the reward system. So they also make a claim there that there's a causal link.
Okay, yeah, that's good. I'll look into that. Okay, just one final question, very short one. In the slide of the meta-analysis that you had, who were the participants in those studies?
That's a meta-analysis of functional neuroimaging studies. I don't recall the exact number of participants, hundreds, I guess. I made an update of this analysis recently. Now it's over 40 studies and with hundreds of participants. We didn't measure them ourselves. It's a meta-analysis of all functional neuroimaging studies that had been published to that date when I did that meta-analysis.
Yeah, thanks a lot. Thanks. Once again, thanks a lot, Stefan. It was a really interesting study. So that's it. Now we go to the next speaker.